Hello everyone, welcome to Easy Life Science, where you will learn science easily. In today's lecture, we will learn about the transgenic plants. What are transgenic plants? Why they are important? How to produce transgenic plants commercially? Methods to produce transgenic plants. Vector-mediated gene transfer. Agrobacterium-mediated gene transfer. 2. Direct method or vector-less method of gene transfer. Physical methods of gene transfer. Electroporation. Bialistic or gene gun. Micro-injection. Liposomal-mediated gene transfer. Silicon carbide fiber-mediated gene transfer. Chemical methods of gene transfer. D-dextran-mediated transfer. Polyethylene glycol-mediated gene transfer. Transgenic plants. Transgenic plants are plants that have been genetically engineered, a breeding approach that uses recombinant DNA techniques to create plants with new characteristics. When a gene of desired character from one plant was incorporated into the DNA of another plant through the process of gene transfer, and that plant is known as transgenic plants, in simple genetically modified plant. Okay then, why these plants are important? What commercial value it has? 1. Resistance to biotic stresses. Through this technique, plants which can resistance to biotic stress such as insect, virus, fungal and bacterial disease resistance can be produced. 2. Resistance to abiotic stresses. Through genetic modification, plants which are resistant to abiotic factors such as temperature, herbicide resistance, glycophosphate resistance, tolerance to water deficit stresses, tolerance to osmotic stress. 3. Improvement of crop yield and quality. The quality of the crop is dependent on a wide range of desirable characters. Nutritional composition of edible parts, flavor, processing quality, shelf life, etc. can be improved. 4. Transgenic plants with improved nutrition. Genetic manipulations for improving the nutritional quality of plant products are of great importance in plant biotechnology. Examples: golden rice. 5. Commercial transgenic crop plants. Transgenic plants, potato and cotton, were, for the first time, made available to farmers in USA in 1995. By the year 1998-99, 5 major transgenic crops, cotton, maize, soybean, canola and potato, were in widespread use. 6. Transgenic plants as bioreactors. Another important application of genetically transformed plants is their utility as bioreactors to produce a wide range of metabolic and industrially valuable products. Let's move to the next step. How we can transfer a gene from one plant to another. What are all the methods are there? We can transfer gene by 2 ways. 1. Vector-mediated gene transfer. 2. Direct or vectorless DNA transfer. First method, vector-mediated gene transfer. Vector-mediated gene transfer is carried out by using a vector which acts as carrier either by agrobacterium-mediated transformation or by use of plant viruses as vectors. Agrobacterium-mediated gene transfer. Agrobacterium tumefaciens is a solborn, gram-negative bacterium. It is rod-shaped and motile, and belongs to the bacterial family of Rhizobiaceae. Tumefaciens is a phytopathogen, and is treated as the nature's most effective plant genetic engineer. There are mainly 2 species of Agrobacterium. I. Agrobacterium tumefaciens that induces crown gall disease. 2. Agrobacterium rhizogenes that induces hairy root disease. How tumefaciens cause crown gall disease? Agrobacterium tumefaciens infects wounded or damaged plant tissues and induces the formation of a plant tumor called crown gall. The entry of the bacterium into the plant tissues is facilitated by the release of certain phenolic compounds like acetosyringone, hydroxyacetosyringone, by the wounded sites. Crown gall formation occurs when the bacterium releases its T plasmid, tumor-inducing plasmid, into the plant cell cytoplasm. A segment, of T plasmid, referred to as tDNA, is actually transferred from the bacterium into the host where it gets integrated into the plant cell chromosome. Thus, crown gall disease is a naturally evolved genetic engineering process. Several dicotyledonous plants, dicots, are affected by crown gall disease, e.g. grapes, roses, stone fruit trees. Tumefacients cause tumor through plasmid called as tumor-inducing plasmid or Ti plasmid. What is Ti plasmid? Ti plasmid is approximate size 200 kilobits each, exist as independent replicating circular DNA molecules within the Agrobacterium cells. The tDNA, transferred DNA, is variable in length in the range of 12 to 24 kilobits, which depends on the bacterial strain from which Ti plasmids come. The Ti plasmid has three important regions. 1. tDNA region. 2. Virulence region. 3. Opine catabolism region. tDNA region. This region has the genes for the biosynthesis of auxin, cytokinin and opine, and is flanked by left and right borders. These three genes are referred to as oncogenes, as they are the determinants of the tumor phenotype. Virulence region. The genes responsible for the transfer of tDNA into the host plant are located outside tDNA, and the region is referred to as Vir gene or virulence region. Vir region codes for proteins involved in tDNA transfer. At least 9 Vir gene operons have been identified. 3. Opine catabolism region. This region codes for proteins involved in the uptake and metabolisms of opines. Apart from the above three, there is region that is responsible for the origin of DNA replication called as IRI region, which permits the Ti plasmid to be stably maintained in Agrobacterium tumefaciens. How this bacterium gets attached to the plant cells, how the Ti plasmid gets transferred to host cells. What are all the mechanisms involved in it? The first step is signal induction from the damaged tissue of plant cell to Agrobacterium. The wounded plant cells release certain chemicals, like phenolic compounds and sugars, which are recognized as signals by Agrobacterium. The signals induced result in a sequence of biochemical events in Agrobacterium that ultimately helps in the transfer of tDNA in Ti plasmid. Once signal got recognized by the bacterium, it needs to get attached to the host cell. The Agrobacterium attaches to plant cells through polysaccharides, particularly cellulose fibers produced by the bacterium. Once bacterium attached, results in production of virulence proteins. As the signal induction occurs in the Agrobacterium cells attached to plant cells, a series of events take place that result in the production of virulence proteins. To start with, signal induction by phenolics stimulates Vir, which in turn activates, by phosphorylation, Vir C. Production of tDNA strand. The right and left borders of tDNA are recognized by Vir proteins, results in the production single-stranded tDNA, ssDNA. When bacterium proised ssDNA. The ssTDNA, Vir D2 complex in association with Vir G is exported from the bacterial cell into the host cell. Transfer of tDNA into plant cells and integration. The tDNA Vir D2 complex crosses the plant plasma membrane. In the plant cells, tDNA gets covered with Vir E2. This covering protects the tDNA from degradation by nucleases. Vir D2 and Vir E2 interact with a variety of plant proteins which influences tDNA transport and integration. once tDNA gets incorporated into the host cell genome, results in formation of tumor. But in case of genetic engineering, the gene of interest is placed in place of tDNA region. When this gene transferred to host cell, the desired character gets expressed. Cointegrate vector. In the cointegrate vector system, the disarmed and modified TI plasmid combines with an intermediate cloning vector to produce a recombinant TI plasmid. Production of disarmed TI plasmid. The tDNA genes for hormone biosynthesis are removed. In place of the deleted DNA, a bacterial plasmid, PBR322 DNA sequence is incorporated. This disarmed plasmid, also referred to as receptor plasmid, has the basic structure of tDNA, right and left borders, virulence genes etc., necessary to transfer the plant cells. In the intermediate vector, the gene of transfer is placed in multiple cloning site, which later gets incorporated into tDNA region of TI plasmid. The co-integrated vector provides several advantages over normal vector. Hairy root disease of Agrobacterium rhizogenes, RI plasmids. Agrobacterium rhizogenes can also infect plants. But this results in hairy roots, and not crown galls as is the case with tumefaciens. The plasmids of rhizogenes have been isolated and characterized. These plasmids, referred to as RI plasmids, RI stands for root-inducing, are of different types. Some of the RI plasmid strains possess genes that are homologous to TI plasmids such as auxin biosynthetic genes. When RI plasmids were infected into the plants, the hairy-like structures were produced around the infected area. Vectors of Agrobacterium rhizogenes. As it is done with tumefaciens, vectors can be constructed by using rhizogenes. These vectors are alternate strategies for gene transfer. Importance of hairy roots. Hairy roots can be cultured in vitro, and thus are important in plant biotechnology. Hairy root systems are useful for the production of secondary metabolites, particularly pharmaceutical proteins. We learnt about Agrobacterium, TI plasmid, how it infects plants and all. Then we can use this concept for transformation of plants. Agrobacterium mediated technique is the most widely used for the transformation of plants and generation of transgenic plants. Before starting transformation, we need select the explant or plant cell. And those cells need to have certain characteristics, and those are: either explants of the plant must produce phenolic compounds, example, autosyringone, for activation of virulence genes. Transformed cells tissues should be capable to regenerate into whole plants. Okay then, how transformation will take place in vitro? If you understand the previous slides of Agrobacterium, this will be easier. First, it needs to develop Agrobacterium carrying the cointegrate vector with the desired gene responsible for desired character. Step 2. Identification of a suitable explant, like cells, protoplasts, tissues, calyces, organs which must possess requirements described above. In Step 3. Co-culture of selected explants with prepared Agrobacterium, where bacterium infects the cell and causes transformation. Followed by killing of Agrobacterium with a suitable antibiotic without harming the plant tissue. Once Agrobacterium infects the plant tissue, it is important to eliminate the bacterium from the culture using suitable antibiotics. Once bacterial cells were killed, the transformed cells were selected and transferred to suitable culture media which supports the growth of shoot and root or the plants.